Hi everyone, I am Rita and I'm a first year PhD student at the University of Sapienza in Roma, and I'm going to present a planned project on the processing of body-driven conflicts of images in anorexia nervosa. Okay, there's a bit of a big delay on the slide change, but I will keep on proceeding. So to begin, I will refresh on what is cognitive control. That is an important human function to guide our behavior until specific goals. And for this, we usually have selective information processing. We monitor our performance, and we also inhibit distractors. And within cognitive control, we know that there is conflict and error processing, which are two mechanisms that allow this control to happen in our behavior. And so we adapt, we do new tasks, and we regulate our behavior according to the demands. And this has been mapped in the brain, especially in the prefrontal cortex and the anterior cingulate cortex. In experimental psychology, this function has been studied with a lot of different tasks, but one of the tasks is the flanker task, where usually participants are presented with the classical way with letters or arrows, with the congruent or incongruent stimuli, and for example, here in the incongruent with SSHSS, and the important thing is that this can cause an flanker effect, which I think you know, but is that participants reply slower and more incorrect on these trials, and so there is a neural response to this conflict, which you can see on EEG with the specific markers, has N2, the theta frequency band, and the error-related monitoring, which I will explain later again. And yes, unfortunately, this important cognitive control that humans have is disturbed in several psychiatric disorders, as in depression, anxiety, and anorexia nervosa, which is the one we will assess. And I find interesting because in this psychiatric disorder, patients usually have, or not, but it depends on the type of anorexia, but hyper-controlled behaviors. And this is usually related to patients that have a self-representation of distorted body image. And the combination of these two usually leads to later on the metacognition to influence on other cognitive functions. And so due to these hyper-controlled behaviors, in literature, we can see that the patients have a better performance in the flanker task than in controls. And this has been found with classical stimuli as the letters, but one of the researchers in our lab some time ago created this task with the bodies there, where they did the same flanker task but with the images of the bodies, and he found that in this task actually the patients perform worse than the controls. So they have more conflict being processed than in the letters, and they perform overall worse than the controls. So from this behavioral study and other results in flanker studies with anoretic patients, we wanted to understand better what is happening in the brain, and so what are the neuromechanisms in this specific task that code the image processing and also the conflict and error monitoring. So this is our aim, to look at the neuromechanisms in the anoretic patients. And to conduct this study, we will recruit 24 controls and 24 patients. And in the first session, we will do questionnaires to pre-screen these patients for having the symptom of body image distortion, because not all anoretic patients have it. And then on the second session, they will come to our lab and they will do the body flanker task and another task, which is the body discrimination task. During the body flanker task, participants will have the continuous EEG recording, and the body discrimination task will be done after the flanker to validate that the participants have this procedural difference between the two silhouettes. They can actually differentiate them. So here is the flanker task, and we do one block of the upright and another inverted, and we do the inverted condition to control the upright observation, and participants have to reply as quickly and accurate as possible, and we will perform this both conditions 4 times, 4 blocks. So in total there will be 480 trials, and they are 4 conditions. So 2 congruent and 2 incongruent, 1 with the overweight target image flanked by the underweight, and 1 with the, did I say the overweight? The overweight target by the underweight and the underweight target by the overweight. And in the EEG, okay. I think now it passed. In the EEG, we will first look at the visual processing of the body silhouettes as the target. So here, and we will look at the usual ERPs related to image processing, but also attentional resources and perceptual processing, as the P1, the N190, the P2, and the P3. And for the performance monitoring, we'll look at the conflict and error processing, the ERP N2 that is there, that is associated with the conflict monitoring, and the theta frequency blend, which is between 4 and 8 Hertz, and has been also associated with the different powers in specific conflict situations, and also error. And for error processing, we will look at the event-related potential, the error-related negativity, which we will also hope to analyze over time for performance and monitoring modulations. But in very short, we have the behavioral measures has reaction times, accuracy, and performance, the performance index, and these ones are the neural indices, and then we will apply mixed ANOVAs. And we will also try to correlate our dependent variables with the psychiatric scores from the questionnaires. And this is just a planned project. I just entered this project, but we hope to find, based on previous results from the study in our lab, but also other studies with the food images during the flanker task, that this specific condition of so the central target being the sort of a positive stimuli, and the target flanking the central target being more emotional arousing, that patients will have more conflict processing. So there is more conflict and they have to allocate more cognitive and affective resources, and so the performance will be worse during this specific condition. But also comparing to controls, we hope that in this task, no, we don't hope, but we think that in this task they will perform worse than the controls, which is different from the usual classical flanker. And for the EEG, we think these behavioral results will be mirrored in the EEG, so that there will be greater amplitudes of the specific conflict and error monitoring ERPs, which will reflect more conflict responses. And so our next steps is to collect the data to hopefully identify some EEG markers and what are the differences with the controls, and that this study is also done. So maybe we can in the future inform studies with neuromodulation. And I want to thank you for hearing my talk, and for all the team that has participated in this project, especially my supervisor. And if you have any questions, you can also email me. Thank you.